For those who do not know, Rachel Armstrong is a professor of experimental architecture in Newcastle University. And she also has a role now this year in the studio with some colleagues, Jolik and Snelle Janssens and Annelies de Smet, in which she works with students from our institute. Why I think it's important to have Rachel Armstrong with us tonight, and hopefully later at other occasions too, is that it's someone to listen to with utmost attention. Because she challenges, in my opinion, a way of thinking that still pervades architecture and the discipline of architecture, and that has done so for a very long time. What I refer to here is the classic dichotomy between matter and form, in which matter is seen as inert, and form, on the other hand, is both an ideal first, but also, and I think in this context more importantly, something that we willful humans impose unilaterally on matter. And in that model, matter doesn't have a big role to play. Rachel Armstrong, in my opinion, in my feeling, proposes another take in which matter is treated with more generosity, and from a perspective in which matter is vital and vibrant, something living rather than dead. She's also important because she embraces complexity as a relational field that is shaped by reciprocal generative interactions between the human and the non-human, and in which the non-human is considered at least as important and at least as active, I think, as the non-human. That's my personal interpretation of listening and reading. She also defies analytical thinking that parses the world in individual entities and emphasizes a view on agency that is congregational and collective. Working together with is then the way to go, and as a replacement for an attitude of unilateral imposition by means of force. Finally, Rachel Armstrong does not limit herself to point at issues or problems that need attention, but she also acts on them from a designerly point of view, speculating, experimenting, looking forward to a better future, and helps us to do the same and act together with her, hopefully, for the better future. Thank you, Rachel Armstrong, already for your talk, and I leave the floor to you now. Thank you.
Thank you, Thierry. Thank you, Lilette, for those wonderful introductions. And I'm actually really excited to be speaking to you all today. I'm going to try an experiment. I'm going to talk about a subject that I've not spoken a lot about before. And it's really been in conversation with people at KU Leuven to actually think about experiment, think about what comes next after the strange time that we find ourselves in. So we're ready to go here. Out of the Anthropocene's cauldron of pollution, animal welfare abuses, urban overcrowding and international travel, steps COVID-19. This novel coronavirus, a highly contagious type of human and animal pathogen originating in bats, is a deadly new neighbor. Spreading through direct contact and indirectly through shared surfaces like door handles, it moves rapidly through cities, mutating and shape-shifting as it goes, maintaining its pathogenicity. From its origin in the wet markets of Wuhan, sometime around November 2019, this deadly RNA virus spreads globally within months, infecting more than 4 million people today. Some remain asymptomatic, whilst others have succumbed to a spectrum of illnesses, including a devastating viral pneumonia. And to date, It's responsible for over 280,000 recorded deaths worldwide. This unbelievable event was not unpredictable. A recent string of epidemics, from HIV in the 1980s, SARS in 2003, H1N1 in 2009, MERS in 2011, and Ebola in 2014 to 2016, means that we should have been amply prepared for this crisis. In December 2014, President Barack Obama warned that the United States needed to prepare for an upcoming pandemic. In 2016, exercise sickness in the UK simulated the impact of a hypothetical H2N2 influenza pandemic. And as recently as 2019, Event 201, a high-level pandemic exercise conducted by the Johns Hopkins Center for Health Security in partnership with the World Economic Forum and the Bill and Melinda Gates Foundation hosted, was held on October 18 in 2019 in New York. But like the challenge of climate change, we seem to know what an effective response entails. But in practice, the inertia in mustering a timely response has taken a devastating toll. While we learned so much about what viruses are and how they reproduce and spread between the H1N1 pandemic in 1918 and the onset of COVID-19, our metaphysics of viruses and of ourselves in relation to them remains as false and as inadequate now as it has always been. Now, from a biopolitical perspective, which goes beyond Michel Foucault's notion of human subjectification by institutions, an understanding of viruses can be formulated by a more than human perspective, or what Maria Puig de la Bellacasa calls an alter biopolitics, asking who or what might be considered a proper part of a mutually dependent world. Now, this is especially important for viruses because they uphold an outsider status on the cellular tree of life. Now, the etymology of virus itself is derived from Latin, inferring a malignant fertility, combining the toxicity of snake poison with the potency of animal semen. Throughout the Middle Ages, it was equated with contagion, but was only described as an infective particle in the 1930s, following the crystallization of the causative agents of an economically catastrophic blight in tobacco crops. Now, through the 20th century, the degenerative status of viruses has been compared negatively with real life. Regarded as mere infective agents, these depictions bear similarities with the absences attributed to female characteristics compared with normative male baselines. Consequently, viruses are not considered as things in themselves, but in relationship to the economic and health effects they have upon real life forms. So a more than human understanding of COVID-19 invokes the microbiome, a landmark insight in the field of ecology in the late 20th century that describes how oceanic and forest ecosystems are associated with complex microbial communities. Following advances in biotechnology at the turn of the millennium, which enabled the emerging science of metagenomics, it was suddenly possible to see microbial ecosystems within our personal spaces and our habitats. Now, the extent to which microbes shaped our lives was piecemeal but revelatory, where our physical entanglement with them changed overnight from just a few to literally hundreds of species. Not only could we understand what kinds of microbes lived alongside us and within us, but we could also observe their diversity and begin to understand how they behave in different settings. Now, embodying the collection of all the microorganisms living in association with the human body, the human microbiome boasts a ratio of 1.3 to 1 microbes to human cells. The anthropos, in this case, is no longer the central figure around which the biological universe is organized, but from a microbiotic perspective, is a mere host for microbial activity, carefully sculpted by prokaryotic ancestors. Consequently, what it means to be human has changed. People are no longer isolated bodies, but are entangled with many others: bacteria, protists, fungi, archaea, and viruses. Most of them are commensals. Some are symbiotic, and about 1,400 are pathogenic. Now, while this seems like a large number, they actually account for less than 1% of the total number of microbial species on the planet. So, overturning any notion of the purity of human identity, our newly discovered bodies as ecosystems are vectors for the distribution of the microbiome in the social realm. And more than a question of biological identity, the entangled choreography between microbes and humans forms a natural laboratory for shaping our lives and well-being. Priming our immune system, it informs our rituals of hygiene, influences our health, and modifies our genetics. Within the human microbiome, the community of viruses known as the virome comprises the most abundant and fastest mutating genetic elements on Earth. Unlike other members of the microbiome, the virome doesn't just colonize host surfaces, but also inhabits tissues, where through its rapid evolution, it can hack our genes, even when there's no symptom of disease. Playing an active role in the cellular functions of our bodies, like training our immune system and regulating protein expression, the virome scripts the story of human survival and adaptation. Retroviruses play a key role in this process, as they replicate by turning their RNA genome into DNA, writing themselves into our chromosomes. Blurring the boundary between human information and virus, around 8% of the natural human genome is composed of ancient viral fossils, which have left their genetic payload behind. The best known of these domesticated viral genes are the syncytins, which were produced by ancient retroviral infections that catalyzed the formation of the placenta. In other words, in deep time, viruses have not just shaped the evolution of people, but also enabled the evolution of an entire class of animals, mammals. Now, not just bodies, but also all surfaces have characteristic microbiomes, which make up the microbiome of the built environment. Now, the relatively frictionless exchange of biological materials within the microbial commons, a kind of public space of microbes all around us, forges critical links between humans and their environment right at the point where meaningful exchange occurs, forming a kind of microbial economy. People therefore maintain, disseminate, and even propagate that activity, playing a critical role in establishing microbiome complexity by being important vectors for microbes within the built environment, where 90% of our daily lives are played out. So rendering this idea of the pure human subject at the center of architectural design as obsolete, we are now compelled to understand what kinds of microbes we are living alongside. We need to recognize their diversity and pay attention to how they behave in different settings. Now, the present knowledge of the microbiome of the built environment is incomplete, and our understanding of its nature is therefore still evolving. It's at its earliest stages of understanding, and state-of-the-art includes its mapping and characterization. Heralding the advent of design and human inhabitation for a microbial era, though, the human body, along with its associated microbial ecologies, become part of the solution to challenges like the COVID-19 pandemic, which cannot be addressed by humans alone, nor by individuals. No matter how big or small infectious diseases are, they are subjects for collectives, where appropriate design can re-articulate our behaviour and contribute to the maintenance of health and well-being. Additionally, this notion of being at war with germs, like Emmanuel Macron's rousing speech to the nation that declared the enemy is invisible, elusive, and advancing, dates back to the reign of hygiene, typical of modernity. And it's no longer an appropriate strategy because the enemy is also us. So in the absence of a vaccine or cure, our best response to managing the spread of pandemics is to consider our own microbiomes as part of the problem and the solution. Through an inclusive politics characterized by co-inhabitation rather than alienation, ways of taking care of each other through our conscious engagement with communities of microbes can be established, even at a time of crisis. By embracing responsibility for our actions and accepting our non-innocence in the propagation of disease, we are re-empowered to act by changing our behaviour and conducting ourselves in ways that are fundamentally community building, despite physical distancing and isolation measures. In acknowledging our role as vectors that shape the microbiomes all around us, the care of our communities is no longer delegated to centralized powers, which entrench our passivity in managing outbreaks. Instead, awareness and knowledge of our relationships to microbiomes means that by taking the characteristics of virulence seriously, washing our hands often and not touching our faces, we can adopt and alter biopolitics that redistributes power within our holobiontic communities, not just during a pandemic, but also during untroubled time. With no effective vaccine to prevent its further transmission, the role of the human body is central to managing the virus in the immediate present by addressing the movement of the virus within indoor and outdoor spaces. Now, the scale of disruption is profound, causing massive destruction to human lives, not least by spotlighting inequalities in our societies. Already, it's changing the way we think. Those that can afford to retreat indoors, trying to keep our microbiome safe from invasion through social distancing. Protecting ourselves from brief outdoors exposures, social distancing helps us move through urban landscapes. However, indoor spaces with prolonged exposure to limited air exchange and lots of people are the most likely places to afford infection, and our concepts of risk are distorted. The main sources of infection are being at home, the workplace, public transport, social gatherings, and restaurants, which account together for 90% of all transmission events. In contrast, shopping appears to only be responsible for 3 to 5% of total infections. And as gregarious creatures, we seek online connection in our domestic fortresses, sharing reassurances and anecdotes that assert our humanity. Our routines overturned and livelihoods threatened, we inevitably reach troughs in our ability to cope with a sudden change in lifestyle and sporadically turn on one another. As we reconsider everyday aspects of life, from air travel to going to meetings that involve human contact, all sorts of interactions, habits, and behaviours that previously seemed innocent are now rude or ominous. Outside, we can no longer trust the safety of the public realm that is newly clouded in danger, which, without cognitive dissonance, cannot now be ignored. The critical change in attitude between an invasive microbial disease and our participation in designing the microbiome is the degree of responsibility we hold both in the sense of our wrongdoings that cause imbalance, and also through our need to mutually coexist along with our microbial ecosystems. Containment only becomes possible when people stop acting alone and collectively adopt fundamentally ethical behaviours that emplace our bodies in a geography of awareness of how effects circulate between us and others. An understanding of ventilation systems, surfaces, moisture, bodies and behaviour enables us to collectively act within our living spaces as an immune system. In an age of antibiotic resistance and climate crisis, we're all part of this biological, architectural, immunosurveillance system, and operating through collective bodies, leading collective lives with other collective bodies, many of which are not human. And this helps us make important decisions about how we act. Now, as an amplifier of our more than human biopolitics or autobiopolitics, the range of technologies can help us actively negotiate livable relationships. The COVID-19 pandemic is not merely dealt with in laboratories and hospitals, but tackled in our homes, communities, and cities. Through appropriate intervention, ways of maintaining healthy individual and collective microbiomes can be established. Stress-releasing dance routines whilst taking out the garbage, balcony concerts, and innovative fabrication of personal protective equipment. Such appropriate, smaller-scale actions can empower citizens and bolster morale in the face of massive uncertainty. Critically, local action can operate far more rapidly and effectively than governmental, institutional, commercial, or international bodies. Now, while I have not worked with viruses directly, which once more highlights their challenging autobiopolitical status, I will briefly discuss a range of projects that I've been involved in as exemplars of designing with microbiomes, which really is the task that we have in order to negotiate these autobiopolitical relationships, both spatially and through our communities. So the first I'm going to share is Living Architecture, which is an EU-funded project, and it takes the form of an interior partition wall about the size of a large bookcase. You can see it here in the picture. And the microbial apparatus is suitable for installation in a bathroom or kitchen. Now, it's composed of modularly stacking brick-like components which provide individual apartments for different kinds of bacterial communities. You can see the little bacterial liquids or the microbial liquids in green in the picture. Since each houses a different colony of microbes, their ordering and spatial relations influence the metabolites produced, so the kinds of substances that these microbes secrete. And when they're brought together in these spatial relations, they're known as metabolic apps. Now, so how does this thing work? So as a living structure, we can think of it working like a cow stomach. So we can take wastewater and liquid, and we can pass it through the bioreactor, which is represented in the form of the cow here with many different kinds of stomach. And as it receives this liquid waste from the household, the bacteria living in the stomachs transform it into a range of different substances. So in this particular case, we can take wastewater, light oxygen and carbon dioxide and put it through the bioreactor, and we will get electricity, we will get water, and we will get inorganic phosphate reclaimed from detergents. And so you see that when we create spaces and houses for microbes, we can actually start to choreograph and negotiate new relationships with them. So these events are taking place in little bricks. You saw those in the first slide there. And this looks like a complicated slide, but it's very simple. It's a house for genetically modified microbes. And the reason we'd want to modify them is because we want to persuade them to do things that they normally wouldn't want to do. In this stage of the Living Architecture project, we're now starting to see just how far we can design with a metabolism. So what you're looking at is a house for genetically modified microbes. In the top floor there, you can see a population of microbes that are engineered to overproduce sugar. So these are Charlie and the Chocolate Factory, and they produce heaps and heaps and heaps of sugar. And this sugar trickles through the floor plate and into the basement there, where they have different neighbours. Now, these neighbours don't normally live together. They normally work on their own. But when we give them enough sugar, they will actually start sharing information, and they will start to produce new molecules. And not only that, we can also design metabolisms across different microbial bodies. So it actually increases the range of different processes that we can achieve in these kinds of apparatuses. So back to the actual brick itself. So you can see here a couple of chambers that the light is passing through, and those contain a particular kind of algae. The one that you can't see in is a microbial fuel cell, and that's producing bioelectricity. So we can actually work with different microbial populations to program these into different kinds of metabolic apps. And the idea in the longer run is really to have these inside our homes working like organs, let's say. So let's replace your fossil fuel burner and give it a metabolism so that the actions of the organisms within those spaces, so the little cities for microbes, can actually produce useful substances that provide resource for our home. And so what we're looking at here is a scenario for apartment block in Europe. And the idea is that we've replaced the fossil fuels with these microbial organs. And what we can start to interrogate here is really to rethink about the infrastructures of living. So first of all, we need more water. And the other thing that we need to think about is, do we actually need 230 volts in every home? Can we actually deal with 12 volt systems? Because that is something that the living architecture apparatus using microbial metabolisms can actually provide us. So then can we actually start to reconfigure our daily lives and reduce the amount of fuel we make, recycle our water, and really rethink about what the activity of daily life actually entails. And so you can see in this part of the video, we're getting much more focus then on our relationship with water inside our microbial homes. And these are organs that will allow us to recycle our wastewater and really create these kinds of holobiontic communities where our care of our microorganisms is actually good for our home, is good for our lives, it's good for the microbes. So happy microbes and happy human inhabitants. So that was the Living Architecture project. And one of the things that we realized in this project was really, how do we know how the microbes are doing? If we're going to actually establish a relationship with microbes, how are we actually going to figure out what they need and how well they're doing? Because we can look at slime if we like, but we don't really have a relationship with it. This is the first, let's call it a post-human apartment that we made. On the left here, we have a microbial organ that's entangled with a set of electronics and screens, and the microbial fuel cells are actually producing power for this apartment space, which is called 999 years, 13 square meters. The future belongs to ghosts, and it's a collaboration with Cecile B. Evans, also the University of West of England and Newcastle University. And the collaboration here is really trying to think about what a home is in the future, and do we actually need a person right in the center of it? But back to this idea of how we might start to communicate with microbes. This is the ALICE project, and this imagines a near future time where different bioreactors are replacing fossil fuel systems, and what we're doing is setting up a biodigital interface so that our microbes are living on these microbial fuel cell interfaces, and we can actually see how well they're doing by the amount of electricity they produce. And so ALICE is an installation that helps us start to interrogate what it might mean to inhabit a microbial city. And so I'm going to play this promo movie that we made for Alice, which will start to give you an idea of what a microbial city and interface might look like. So yes, I just wanted to thank the Alice team for that. So essentially, in order to bring something like that to life, you know, a large team, European grant is needed for that. And the idea really is that we're starting not to just be receivers and hosts for microbes, but actually start to negotiate these autobiopolitical and kind of microbial economic conditions, so that we don't just overlook or try to go to war with germs, but we're actually establishing new kinds of interfaces through which we can have meaningful exchange. And this is the last project that I'll show you, which is really still a proposal. It's been submitted to the EU for consideration. Hopefully we'll hear sometime this summer. But this is really trying to scale up microbial interactions to produce urban effects. So effectively, this is like a sewage plant, but what we're doing is we're bringing together wastewater gardens, functional wastewater gardens, where the microbiomes of plant roots process organic waste and water, and create a lovely range of plants. So it's really like a garden, a public space, and bring that together with the microbial fuel cell. And the idea is actually gardens within our city then can actually become power plants in some way, more ways than one, both the plants and also sources for generating electricity. Now, this isn't 230 volts. These will be enough enough power to charge a mobile phone, to make LED street lighting possible. So kind of low power electronics. And I think it's really important because I think that we are looking at an age of low power electronics, which might help us regulate some of these additional problems that we have coming with climate change, because of course all of this is related. The COVID-19 pandemic and climate change are not separate issues. So crediting the incredible team here that applied for this grant, so fingers crossed with that. But I just wanted to say that it does include KU Leuven, led by Nell Janssens, and also the work here really is looking to think about reconfiguring the relationship between technology, people, environment, and these ideas of circularity, finding new technologies that might help us perform circularity in a vastly expanded range of ways. So no aspect of working with microbiomes at any scale is fully resolved right now, and all of them remain problematic. However, these merging technological platforms raise pressing and broader questions about how we live, our ethics, values, economies, biopolitics, and longer-term relationships with nature. The unbelievable situation that we're faced with today is not a one-off event. Like climate change, a pandemic is essentially a man-made disaster, born of poor health and sanitary conditions, the abuse of natural systems, and the opportunities afforded by the growing interconnectivity of a globalized world. Such outbreaks will be more frequent, severe, and costly in the months and years ahead. Should the pandemic reach a pause, our focus will necessarily turn to the other aspects of climate change. In the immediate and longer terms, our present and ongoing crisis means that design must lead the way in establishing new kinds of creativity, intelligence, and care. Heightened awareness of the more than human realm is critical for our personal and environmental well-being. Taking the expanded autobiopolitics of climate change seriously can enable effective action at every front line of disarray where it is most urgently needed. Of course, a new awareness is no panacea, and local actions do not circumvent the role of institutions, but spotlights their role as administrative systems rather than protective structures. In the meantime, design-led research can help us orient ourselves within this volatile era of climate crisis, where effective community actions can hold equal footing with scientific developments, not as a binary choice of approach, but as mutually complementary actions. In the face of massive uncertainty, design-led research and practice can assemble effective responses to irreducibly complex or wicked problems. Such an approach requires an expanded knowledge set or inter- and transdisciplinary experimental attitudes, and importantly, a decentered notion of humanity, where our species is not the beginning and end of all outcomes, so that we develop an idea of design that upholds an ecological ethos. Paying attention to these invisible landscapes and asking questions about where our responsibilities and opportunities lie, we can uphold properly functioning social lives and make lasting differences even in the face of contagion. Going forward, such forms of inquiry and methods for identifying where meaningful action can be taken are vital for addressing the world's larger issues, not as a panacea, but as a valid approach in the absence of a livable normal. Thank you.
Thank you, Rachel. Thank you so much for this fantastic talk and this world you bring us into. Maybe, Thierry, you can lead the Q&A, and as said, all people can ask their questions in the Q&A.
Yes, absolutely, because the goal is to have some kind of exchange, of course. Because I can imagine that many questions pop up in the sense that it's not what I would say the architectural lecture in which we see buildings and shapes and spectacular spatial interventions, but this level of cohabitation with in between, which kind of challenges another kind of thinking. In that sense, maybe to start, I will ask you a question. Rachel, you talk about the fact that we need to, kind of develop an expanded knowledge set. And that for doing that, we, how to say it, we need to be able to share different kinds of knowledge. I don't know your history exactly, but I imagine, or I think that you have you're not trained as an architect, I think. So at a certain moment, you made a step towards architecture, What was it that motivated you to make that step and to basically reach out to architects and say, well, here is something that we need to pay attention to?
That's a really great question. Thank you for that. And essentially, you know, architects have the tool set that I was looking for. So I'd actually come from medicine, where I was looking at the body in relationship to advancing technologies and looking at the status of people in the face of technology. And I realized the social and cultural and artistic dimensions of that investigation were really absent. And so architecture already had a transdisciplinary, interdisciplinary platform that was probably the only one that I could find that would actually be welcoming of, let's call it, an outside knowledge set. I couldn't have walked into biology or, you know, many other many other disciplines to have the conversations that I subsequently had in architecture. So I was actually fascinated by the subject of architecture and the multidisciplinarity, multifacetedness of the training and the subject matters. I mean, there are no two architects are alike. And so it was I went to the Bartlett first of all. So it was a really rich environment in which to start to have these very hybrid conversations. And really convinced me that this was an incredible research platform. And I then went to do an architecture PhD.
Thank you. I have a question. Good. Christine Munoz.
Okay. Well, let me just first say that this was a fantastic lecture, and it was totally redefining and mind-blowing in terms of what we are and how we define being humans, and at the same time this idea of cohabiting with microbes instead of excluding them from everyday life. But I wonder, are there any kind of safety protocols regarding building and actually living and using these microbe walls, which Rachel showed in the video and in the photographs?
Yeah, thank you for that, Christine. It's really important. As I said in the talk, this is not a panacea. It's an appreciation that we do not own this world, and in fact, our microbial, you know, deep ancestors were here way before we were. What we lack right now is, first of all, you know, we've only really just started to see who we're living alongside, and we still don't know most of the microbial species on Earth. So we really are looking at the tip of an iceberg. Having said that, you know, what we're finding out is, let's say, not unmanageable. I mean, we have been living amongst microbes for a very long time, you know, since we evolved, since at least, you know, maybe even the origins of life, if we go back that far. So what's important about this right now is kind of rereading where we are in relationship to the living world. One of the most important insights, I thought, this century, or last century, was Lynn Margulis, who was looking at, she was working with James Lovelock on the Gaia hypothesis. And she identified microbes as the world makers. In other words, the active components of the earth that made, let's call it, life livable, so that we could have reducing and oxidizing gases together, and we could have a disequilibrium, which meant that our planet didn't just go to sleep chemically, but was still very vibrant. And she said that microbes were the world makers. And so when we think about that, and we think about some of the challenges with climate change, you know, we now have a tool set. We now have an awareness of the microbial realm in a way that we didn't have maybe 50 years ago. And so a better understanding of this realm is absolutely critical. It will provide loads of different opportunities, but it is also important to respect, you know, the potency of the microbial realm, We're still developing languages. So Bonnie Bassler, for example, lovely TED talk. You should have a look at it if you're interested in microbial languages. So that we're starting to learn that microbes actually have languages, and we're learning how to speak them, as it were. And as we gain these tool sets, it's going to give us different opportunities. Now, again, it's not a happy ever after. I think our position on this planet is going to have to be hard negotiated. And I think that we're going to have to be smarter and more diplomatic with the creatures that we share the planet with. And in terms of our buildings, there's wonderful work going on at the University of Oregon at the Biobe Center. And so it's environmental engineers that are characterizing the interior conditions that allow the distribution of microbes. So we're starting to map these spaces. We know that ventilation matters. We know that gravity matters. We know the proximity of certain kinds of activities in different rooms matter, but we don't have the whole picture yet. So it's an incredible space for architectural research. It's an incredible space for architectural design. And there's a brand new tool set. So both from imaging techniques, so if we think about the rise of the computers in the 1990s, we've got exactly the same thing going on with microbes as we start to plot our findings. And then the other part is, well, actually, how do we start to design? Although we've done some, initial prototypes, these are not formal rules. And so I think it's an incredibly exciting terrain. I think there's an awful lot that's still to be known. And I think it's a very important moment, I think, for architecture, because I think that these spaces really can make a big livable difference.
There is another question from someone called an anonymous attendee. The question is as follows: architecture is a static discipline and biology is a very dynamic discipline. I don't know if that's true. How can practicing architects incorporate or cooperate, I don't know, with biological materials today? Or incorporate biological materials today? After COVID-19, I believe the approach will be changed to antibacterial design of things. How can we lead this to a more scientific approach? This last thing is not very clear to me, but maybe focus on indeed the fact that we might end up with being more suspicious than cooperative.
I think that is, I think that's inevitable. I think if we listen to the language of our leaders, we're going to war with germs. I think if we listen to the voice of the scientific community and ecologists, I think we need a very different tactical approach. Now, there are branches of architecture that deal with the biological. So I don't know if you're familiar with the whole field of biodesign. So there are many centres around the world now, from Penn to Kent to, many different kinds of centres that are looking at this, bringing together, as you've mentioned, the dynamic aspects of biology with the more traditional elements of material practice. I think what's important about biodesign at this stage is also that it embodies a philosophy of practice. So it's not really just about making wood and, let's say, carving a nice piece of wood and saying, well, that's organic and therefore it's good. I think the biodesign enables us to have a conversation with the living world and through materials and through space and prototyping that is really critical about how we design. And so there is an ethos. There is, for example, a condition of care for your biodesign materials. Someone like Symbiotica might even say that those materials actually have a status, and if they die, then they need to be treated appropriately. So there are all kinds of ethical questions that get tangled up in these issues, and so these issues are actually being engaged with by design-led communities, artists and designers and architects. And again, it's a relatively new discipline. But I think it's a very important one because it's also helping us find a language for design as well. So I would say that if you're interested in this area, that the biodesign and looking at people that are practicing biodesign is probably a good place to get some inspiration and figure out, maybe some of the choices you might want to make.
There is Vanda here. I think it's one of your students, Lilet, who has a question to ask.
Yeah, I invited you, Wanda, to become a member of the panel.
Hello. I would like to really thank you for the perfect lecture. It was amazing. I was really looking forward to hear it from you. And I would like to ask in behalf of us students, what advice do you have for students of architecture that will soon graduate and enter the job market? What kind of qualities should we always bear in mind, and what should we never do?
Okay. I wouldn't dream of telling you guys what to do. I think what I will say, I'm going to be naughty and I'm going to say, you know what, I would really like to see all architects be active researchers, whether that is formally within a university or whether that is more informally within a practice, because you guys are the cutting edge of change. So, you've really, I'm so excited about you guys because you're at this incredible moment where you have no, let's call it superiors, in the sense of there is nobody doing, let's call it design for climate change at a level that you guys couldn't match with clever thinking, dexterity, persistence and vision. I mean, you really are in a position where you could make very big advances in a very small space of time. What it means, though, you have to stay alert. And yes, absolutely, take the knowledge that your professors are sharing with you. Learn, what the profession is about. But then when you go into practice, you find your own way, kind of assimilate and incorporate and really, develop who you are as a designer and find different ways to engage that passion. And also don't be selfish. Work with others. It's the most rewarding kind of work because you get to share, kind of the joy and the sense of achievement when you're in a team and you're, sharing, ideas and the frustrations, because, not everything works first time, not everything looks great first time, but, being able to engage in a group of people that share an ethos with you, that are equally driven to bring about positive change, to make small, maybe, but important changes that maybe you're not the person, maybe I'm certainly not, that's going to change the face of architecture, but by together, small incremental changes, increase that threshold for a radical breakthrough, and you're part of the tsunami. So I think all of you guys are in that position where you can make a real difference. 50 years ago, someone in your position, I wouldn't have been saying that to you, because the economic situation is stable, the practice is stable, the career pathways are stable. All of that stability is gone. So on one hand, it's frightening because you don't know what's coming next. But on the other hand, there are some incredible opportunities. So having a vision, understanding who you are as a designer, and not being afraid to put some of that out in the world and find your community. And I think you'll have a really fulfilling life in the manner of design that you choose. But definitely research, keep asking questions.
Thank you very much.
What a fantastic plea for architecture non-architects can make, always. Fantastic. Thank you, Vanda.
Can we go for one more?
Yes.
Yes, that's a good idea. Let's continue a little bit. You talked about living architecture and making different microbes work together to generate energy. Do you think that this kind of system could be used on a larger scale in the future? I guess the good thing about bacteria and such, it's as living organisms, they reproduce. So they form a flux of resources and not a finite resource. But what about the infrastructure needed to make this work? That's the question.
Yes, these are essential questions. I mean, and we've been exploring some of these in the Living Architecture project, in the ALICE project, and in the Microbial Urbanism project. And so essentially the infrastructures for microbes are the infrastructures for life. This is what the Biobi Center has been finding out in Colorado as well. So we know that the flow of air, the availability of water and food are exactly what microbes need, just like all of us. And I think there's something very interesting in that, because what it starts to do from an architectural perspective, it invites us to cohabit spaces. So if we are using, let's say, if we're working alongside microbes, or we're co-inhabiting with microbes, and that we want to form a coherent community where we're sharing resource, and where we're producing things that we need, then we need to figure out what everybody needs and how we actually design mutually, including our microbial brothers and sisters, and make sure that they're okay. And that's a very different notion of technology than putting technology to work. And so going back to your question about ethics, I struggled a lot with the genetically modified organisms, because you have to think about whether that's correct or not. and although empathy for a microbe may be a bit of a far stretch, you really need to think into those spaces as to what is appropriate, what are you trying to do, whether this is something that you can uphold in terms of your values. And I got to a stage where I realized that with the microbes that we were genetically modifying, they were so adept at dealing with this extra genetic information that they would spit it out after about 10 days anyway if they didn't like it. And naturally, microbes were used to getting infected by viruses like bacteriophages. So it was part of a system of exchange that they were involved in. And so I felt that it was something that, because we were asking these questions about how far we could stretch this idea of design with metabolism, and how far we could ask microbes to work alongside us, it was worth doing. And what we could find out from that then was, how can we find the natural organisms that perform those equivalent tasks? So it was a bit of a cheat and a bit of a quick fix. But working alongside and, let's say, engaging with technologies that have an inner life in some way, no matter how big or small, I think really does require that ethical position. It does require you to look after them, care for them. And there may even be at a point where we've got our interfaces going and we're starting to empathize and relate to our microbes because we have an appropriate form of communication. So maybe they're like a pot plant, or maybe they're more like a pet. But I think that we will actually develop those kinds of interfaces, and I think that that will help us better relate to non-human others. And I think that's important, not just for microbes, but it's good for our trees and it's good for our pets. And I think that that kind of reconfiguring of our responsibilities and our engagements in a broader society that's not just human is really at the heart of this.
Rachel, I have a question here from someone who, Linda online?
I'm on. It was trying to find Zoom. You hear me?
Yep.
Okay, perfect. So I mainly work with heritage at the moment. So I was wondering, how will it ever be possible to infuse it into old buildings, and how would it work? Because I cannot change a lot about an old building when it's protected. It also has its own story. Can we add actually this new kind of story into it? And will it ever be possible, do you think?
I think the short answer is yes. And I think the questions are those related to design. And if we can figure out how to make microbial urbanism, there's no need to have every single bioreactor inside a specific building. It will really be about piping in, a bit like services and adding services to a historic building. So I think there'll be lots of different ways that we can actually think about how we use microbial materials, technologies, processes in order to better support spaces. So I think it's not like just having to install a particular kind of boiler. I think that because this is microbial life, it's very protean, and I think that through design we'll be able to respond in quite a variety of different ways. So I would hold up hope that it is possible for these kinds of relationships and systems to have a benefit to historically important buildings.
Thank you.
I think we're at the end of the questions from our public. Maybe I still have at least one question. I was wondering, basically what you are talking about is a kind of paradigm shift, in the sense that we have to think about certain things in another way and therefore address them differently, have another attitude and so forth. And traditionally, indeed, I think in architecture, when we refer to the static, there was this kind of aspect of control where you can say, well, these material assembly that we make here will remain like this for a certain amount of time. And so we can kind of assure people that it will stay there for a while and do certain things that we want it to do. Whereas what you now, the way in which you think about it is that instead of working with these this temporal continuity, we need to work with temporal change and with processes as architectural material, which is another attitude. How do you think that we can incorporate this new attitude and working with processes in architectural education?
Yeah, this is a really nice question, because I think that we're used to static thinking, but I think that when we think about processes and when we think about dynamics, actually we start to recognize patterns. So, you know, if we think of fluid dynamics, for example, we know that under certain conditions, certain patterns get set up. So it's really by being more familiar with some of these materials, we can start to make appropriate choices so that if we need a certain behavior or performance on a site, then we may have a range of systems through which we can choose to work with. And sometimes the answer will be a fixed concrete post. But what we hope to do is to be able to expand that portfolio of choice so that not everything is a concrete post. So I think it's like that, and I think that there's a dexterity of pattern recognition. When things are moving, you start moving into pattern recognition, and we're starting to see that in some of the computer art. You know, the algorithms that are able to kind of generate incredible geometries through algorithmic flow. And I think there'll be some overlaps with that. So it won't just all about be, you know, static geometric or so chaotic that we don't know what's going on. I think there'll be all kinds of different degrees of predictability and stability within that.
There was a question about computers. Is it so that that we are now able to model processes, like, and show them and work with them in dynamic ways, whereas before that we were not able to do that? Does that make a difference on putting this on the agenda too, the fact that we have other means?
I think we've got more tools. So some of those tools are digital, and the predictive power of digital tools is going up. We're also looking at the advent of quantum computing, which means that for the first time, computers will be able to start to predict protein folding, which is one of those things that you know, traditional digital computers have not been good at. But also we're looking at forms of unconventional computing, where we are computing directly with matter. And so a kind of new age of analog computing is coming along. So for example, we're starting to see the slime mold organism being incorporated into mapping cities, for example, or detecting nodes on a transport system. So there are different forms of analog computing that are starting to come with advanced materials knowledge. So I think with traditional digital computing, with quantum computing, and with forms of unconventional or material computing, I think there's a powerful tool set for helping us, let's say, configure some of these relationships. And the more we do it, the more familiar we'll become with this, and we'll know what the constraints and limitations are. At the moment, we've got very little idea of where the boundaries are. But as we do more of this, and as this next generation who's been asking questions gets super familiar with the tools, we'll think it's a cinch.
Thank you. I was confused a little bit because I saw a question now which appeared in front of me that maybe if you still have the energy to get it. A question from Ahmed Fatih. I probably pronounce it in a totally wrong way, but it's Ahmed Fatih Tank or T-A-N-C. Lila, do you see?
Yeah, I hear all.
Oh, there you
Hey, Rachel. Yeah, first of all, thank you for great presentation. I have a just quick question. Like when we talk about living with nature and working with nature, we should also think about the death of the buildings or the death, because the process of biodegradability is a part of the nature. So in my bachelor years, I was searching about reverse engineering, that how can we create materials that can be biodegradable through another these processes. Is it possible to regenerate or kind of a mutation process of these microbes that we can just demolish or decompose some materials? Like when we build a building, can we just code these microbes to decompose them?
You know, it's a really interesting question, the idea of regenerative architecture. And one of the things that we haven't mapped is the kind of biochemical pathways of compost. There's a saying that there's more life in a dead tree than there is in a living tree. And that's because of the boundedness of the potential of something that doesn't have a kind of constrained program on it. And so it's, in some ways, there's a metaphor about buildings with that with the right concept of deconstruction, decomposing, and disassembly, we could actually think about regenerative architectures. We're not there yet. So I think this is one of those areas for research, actually, that I'm hoping that you guys really tackle head on, because it's really important that we don't have buildings that are concrete fossils and industrial fossils, and that we find out how we can actually disassemble and reconstruct buildings. That may not necessarily be down to the molecules. There may be a geometric order underneath it. There may be another kind of order, maybe soil, for example. But these are very important questions. Yes, of course. I mean, nature has a cycle of life and death. We've paid a lot of attention to the life part. We've paid a lot less attention to the death taking apart part and then reassembling part. And I would say that is a place for research, which would be very productive.
Thank you so much.
Good. Well, we are at the end of the questions, the written down questions. So maybe, well, I would propose to end the conversation here. I would do it with a sentence that has been written here by the anonymous attendee. You are so inspiring. Thank you for that. Thank you, Rachel Armstrong.